there's a weird little echo. Oh, it's probably because I'm close. So thanks everybody for coming to our second Center for Animal Welfare Science seminar for the term. We're trying to get back into having the center do consistent programming that really is useful to the people who are taking the time to attend. We know your time is very packed, and so we want you to have good return on investment when you come to these things. So today's seminar should be a treat for you. We have with us Dr. Esteban Fernandez-Juricic.
Perfect.
He is a professor of biological sciences. He collaborates quite a lot with faculty in the animal sciences. His work is very broad-ranging. And one of the things I asked him to talk about, because I think it's particularly relevant to this audience, is the importance of interdisciplinary collaborations. Oftentimes we may think we're doing interdisciplinary work when sometimes we're doing work that's just different areas of focus within our disciplines. That's important, but it's really important as well to bring in additional perspectives and insights and tools from other disciplines that can help us ask even better questions and solve problems even more effectively. And then even better yet, have much better uptake and application of the work and dissemination of it. So thank you for taking the time to come today. For those of you joining us online, if someone can signal to make sure you can hear everything okay, we will jump in and get started. And I'm going to make a plug for the next seminars that we have coming up. They're going to be on March 26th and April 28th. Or is it April 30th?
April 30th.
It's April 30th. Sorry about that. Our next seminar is going to focus on the role of AI and ethical considerations in AI and its applications to the work we do in animal agriculture. And our very last seminar is with an amazing and very famous speaker, Dr. Clive Wynne. If there's something you need to know about dogs and dog behavior, that's a different topic. So we're trying to give you a nice spread of topics focusing from animal agriculture to ethical implications and work with other species, including companion animal species, so that everybody has something they can sign up for and be really excited about. So with that, I will turn it over to our speaker.
Thank you. I need to do this out. Yeah. Can you hear me? Excellent. Well, thank you for coming today. So today's talk is a little bit weird from the following perspective, right? So as Candice said, I'm going to try to talk about interdisciplinarity, right? Which is always challenging. And I'm coming from the field of quantitative ecology, right? And in quantitative ecology, what we do is we try to apply mathematical models and statistical models to study the interactions between organisms and their environment, right? And then I'm going to provide some, not even suggestions, some opportunities, right, for collaboration between disciplines. So I want to start with, there you go, with a disclaimer. I'm not here to teach you anything. You are the experts in animal sciences, okay? I'm just here to highlight some things that I have been doing with wonderful collaborators, with the idea of, well, you know, can we use some of these things for some of the projects that you are doing? I want to start by thanking these four individuals, Darren, Greg, Marisa, and Pat Warner from FNR. I've been learning from all of them tremendously about animal welfare. It's not my specialty, right? But slowly I'm getting into that field. I also want to thank Patrice Dampart, who is the research associate in my lab. Whatever I'm going to present today would not have been possible without Patrice's amazing role in all this. All right, so my lab tries to address the question of how visual perception affects behavior, right? That's sort of the umbrella question. And the approach that we use is we first go and characterize the visual system of the study species, right? Using a bunch of different techniques, right? So we measure, we characterize their color vision, their spatial resolution, their temporal visual resolution, and all that. So then with all that information, we use these mathematical models called visual models, right? that try to make predictions about how animals are perceiving visual signals in their environment. And those predictions, then we go and test using animal experiments of any kind that you can think of, right? So that's our overall approach to address these questions. So basically what we're trying to do is we're trying to bridge the gap between visual perception and behavior, right? And our research has had implications for evolution, sensory mechanisms, conservation ecology, and animal welfare, which is what I'm going to be talking about today. All right, so two themes today. The first one is some mathematical and statistical modeling applications to animal welfare as a result of the collaborations I'm having with the individuals I mentioned before. And then I'm going to switch gears and talk about how could we improve the reproducibility of the quantitative research that we do, which is a very important topic these days, given the increase in quantitative techniques that we all have to deal with. And AI is one of the many, right? All right, so bird and animal welfare, right? So there are multiple bird species that are used for in poultry, right? But we also, and these species belong to different families and orders and all that. But we also have to be aware that there is a big picture here, which is that they belong to this group of birds, right? And these days, it varies from year to year. We need to be thinking about 10,000 living bird species on Earth nowadays, right? So the poultry species occupy some places in this huge diversity of bird species. One of the things that you already know, but I'm going to be talking a little bit today about, is that bird vision is fundamentally different from human vision. So they perceive their environment in a very different way from the way we do it, right? And that has to do with many things. Here's a quick list, right? So as you may have noticed, many species have their eyes laterally placed, not all, but many bird species. So they have really wide lateral vision. Their eyes are large relatively to the size of their bodies. And consequently, for their body masses, they have high visual acuity or high ability to resolve something far away. Humans have three types of visual pigments, right? Distributed in three types of single cone photoreceptors. Birds have four types of visual pigments. So the translation is that they see many more colors than we do, and they see colors that we cannot even imagine in our brains, okay? The other interesting thing about birds is that they have single cones like we do, and those single cones are associated with the processing of color vision, color signals. But then birds, humans don't have the double cones. Birds have these double cones that are associated with the processing of achromatic signals and also motion, right? So they have a visual channel devoted to color and another visual channel devoted to achromatic and motion vision, which is pretty interesting. Birds also have high temporal visual resolution, right? So they can process stimuli in their eyes very quickly. And it makes sense because they have to fly around a bunch of obstacles and they need to make sure they don't hit anything. So given all these differences, then we can ask the fair question: would a bird perceive this sort of aviary in the same way as we do? And the answer is no. Not at all, right? Now, so that's an easy answer, right? But the more difficult answer is, well, tell me details. And that's when things get very complicated, right? The details. So the other concept that we need to keep in mind is that, yes, birds and humans, very different visual perception, but within birds, different bird species also very substantial in their visual perception. So this is a study that we published recently, right, looking at one of these visual traits, which is this visual acuity or the ability to perceive detail far away. And what happens is that there is a large variation in visual acuity across different bird species, or at least the bird species that we studied. And that translates into the same stimuli being perceived by different species in very different ways. So with all this background, one of the questions that we pose is, can we design spaces tuned to the eyes of the poultry bird species? So that was a challenge, right? And there are animal welfare implications of doing so, right? Because if we can do that, maybe we can improve the way in which they function, right? The way in which they interact with each other, the way in which they interact with food, the way in which they feel within these aviaries, and also their ability to express normal behavior. The idea is to hopefully reduce aggressive behaviors between animals, reduce the incidence of injuries that they have with interacting with objects in the environment. And maybe if we do that the right way, maybe we can also increase production. So what we decided to do is to apply this sensory approach. That's one of the aspects. We did many other things, but I don't have the time to mention them to you today. And we put together two proposals. One to study the Peking ducks, right, from that point of view, and also to study hens. And we were extremely fortunate to get them both funded through the USDA. And that's something that we are working on right now. So, applications of mathematical modeling into animal welfare. So what I'm going to do within this approach that we're using is I'm going to go a little bit deep into these visual models. What are these visual models? What do they do for us, right? So these visual models come from a rich literature in visual ecology, right? Which tries to study how animals perceive their environment, and based on that perception, how they interact with their environment. So these three individuals pioneered the development and use of mathematical algorithms that would mimic the way the avian retina would process signals, right? And one of the key things about these models is that they take into consideration the visual perception of individual bird species. So in order to apply these models, you have to go and study the visual traits that make a species from a visual point of view very particular. And this is what we call these visual models. You know, some other people call it perceptual models, but they're all the same thing. Okay. So what is the output of these visual models? So we try to understand them from a top-down point of view. So the output is a concept called visual contrast, which is how much an object stands out from the background, right? So this gives you an idea of visual contrast. So imagine that the object that we're trying to model is the red apple, and the background are the green apples, right? So as you can see, you go, yeah, the red apple stands out pretty much. The interesting thing about visual contrast is that it's not object dependent, right? Because we can apply the same concept and get a similar value of visual contrast with this, when we flip this. So the background now are the red apples and the green apple stands out, right? So that's what the model's output is. An estimation, a mathematical estimation of how much the object stands out from the background. What are the components of these visual models? Well, they are summarized in these arcs, a schematic representation, and the main components are the spectral properties of the ambient light, which is the A, the reflectance of the object and the background, right? The transmittance of the light from the object to the sensory system of the animal that is trying to perceive that, which is symbolized by these S, right? So these are the main components of the model, right? So one of the things I told you a few minutes ago is that birds have this achromatic channel and the chromatic channel, right? So this is reflected in these visual models because the model calculates a chromatic contrast and also calculates an achromatic contrast. So it takes into consideration these two visual channels, right? So here we have high, here we have low, and the same on the achromatic scale. All right, so how do they work, right? So I'm going to show you some slides. Don't be scared. Bear with me. We're not going to go into deep math. So the models that we use there are multiple vision models. The one that we use the most is the photon catching receptor noise model, right? And this model has some input parameters, which are the parameters that we have to go and measure from the species that we're interested in modeling. So the first one is called photoreceptor sensitivities, right? And here is some of the components related to the photoreceptor sensitivities. So for you to think about these, think about an eye, right? The light goes through the cornea, the lenses, the vitreous humor, and eventually it gets to the retina. Well, the cornea, the lenses, and the vitreous humor are absorbing portions of that light, right? So that's what we call the transmittance of the light. So we go and measure that transmittance of these components. And then when we have the eye, we cut it in half, and then we extract the retina. So that's a retina that has been flattened. That's why you see those cuts at the edges, right? And then we put that retina under the microscope. We can see under the retina and the microscope in the bright field and in the fluorescent light, right? So you will notice those dots popping up in the image. Well, those dots are components of the single and the double cone. And they're specifically these organelles called oil droplets. So the oil droplets, what they do is when light gets into the cone, light gets in this direction, they filter the light, they have carotenoids inside them, and then eventually after that filtering process, they get into the visual pigment, where phototransduction takes place, okay? So all these things that you see there are these oil droplets, right? And there are different kinds associated with each type of photoreceptor, blah, blah, blah. So then what we do is we go and measure the sensitivity of the visual pigments, which is this, and the sensitivity of visual pigments is usually measured through a property called the lambda max, a lot of lingo, don't worry, which represents the peak wavelength in which that photoreceptor is absorbing most of the light, right? We also measure the absorbance of the oil droplets. So this is what the absorbance of the oil droplet does. And what an oil droplet is doing is absorbing light and in all these wavelengths, absorbs, up to this point in which it lets light go in. So that point is called the lambda cap. And I mentioned these names because I'm going to use them in a couple of slides. So when we put these two elements together, we have the overall sensitivity curves, which also go into these models. So as you can see, it's a pretty involved process to get this model to work. Okay, the second set of measurements that we take are associated with the reflectance of the object and the background. And also, we measure the ambient light conditions through something called irradiance, which are the spectral properties of the light, right? And here's the math. We don't need to pay attention to that. And we do all these measurements with a gizmo called the spectrometer, right? That has a probe, and we use that probe to, if we want to measure the reflectance of something in the imagery, we put the probe next to it, and then turn on the spectrometer, and boom, we have a spectrum coming out of that. The final component of this model is related to the densities of the different photoreceptors, right? And the way you want to think about the densities of the photoreceptors is similar to a old TV and a new TV. So many years ago, we had these TVs that had a very limited number of pixels. So think of the photoreceptors as each of those pixels. So if you turn on one of those TVs, when I was a kid, right? You could see something, but it was blurry and something like that. But these days, you have these 4K or 8K, and you go, whoa, the resolution is insane. Well, the same with the photoreceptor. So some photoreceptors have a low density, right? Which means that their signal becomes very noisy, right, for the animal, whereas other photoreceptors have a really high density, which means that it's a strong signal for the animal. So not all photoreceptors have the same density across the retina. That's the bottom line. Great. So when we have all these things, we input that into this impossible to explain equation, right? And then what the model does is it estimates the distance between the object and the background, okay, in the color space of a bird, which is defined by these 4 different types of visual pigments. That distance is the visual contrast. So the closer this object is to the background, the lower the contrast is going to be. The higher the distance is between them, the higher the visual contrast is going to be. Does that make sense? Okay, great. So when we do that, when we use these models, the models assume there is going to be a magic value, magic because it's difficult to quantify, that we call a threshold. Below that threshold, the animal is going to have a really hard time to distinguish between the object and the background. Above that threshold, it's going to be easier, right? So there are different thresholds used for different species. We use these 4 threshold, and the model spits the output of chromatic and achromatic contrast in units called just node. Probably too much information, but we will use it in a few minutes. All right, so as one of the many questions that we're trying to address in these projects that we're collaborating with is why lighting would improve Peking duck welfare in these barns, right? So, so what we decided to do is to measure many of these parameters in Peking duck, but also compare those parameters with the mallard duck, because the Peking duck is a domesticated version of the wild mallard, and see what's going on there, right? So in terms of visual pigment sensitivity, remember the lambda max I told you before, right? So I'm going to be showing you some figures contrasting that lambda max between the mallard and the Peking duck. So this is for the long wavelength sensitive photoreceptor, the medium wavelength sensitive, the short wavelength sensitive, and the violet sensitive photoreceptor. So one of the trends that we see that seems to be pretty pronounced is that the Peking duck seems to have that lambda max sort of shifted to longer wavelengths, right? So it's always, the wavelength is always, not always, but at least in 3 of them, higher than in the mallard, right? So keep that in mind. In terms of the absorbance of the oil droplets, the lambda cat, right? So here we have a table showing the difference in lambda cat between the mallard and the Peking duck. So if we have a negative value, it means that the Peking duck has a higher value in terms of wavelength, right? So as you can see, there is a trend, in some cases very small, but in other cases more pronounced, for the Peking ducks to have that sort of shifting towards that absorbance towards longer wavelength. So the bottom line is that the Peking ducks seem to be redshifted. So the visual system relative to the mallard seems to be more sensitive to those longer wavelengths, which are basically, the translation is the red color, right, from our point of view. So then the prediction that we can have is that Peking ducks should have a higher sensitivity under long wavelength lights. In other words, Peking ducks should feel more comfortable when they are below a red light than other wavelengths. Does that make sense? But so I'm going to show you 2 amazing videos that Greg shared with me. I love this video, right? In which he did the experiment. He put the Peking ducks under blue light and red light at the same time. You tell me what you want to do, Peking duck. Oops, no. Oh no, what's going on? Sorry about that. It worked before. Oh, there you go. Isn't that amazing? So you see the Peking ducks, exploring and exploring, but at the end of the day, I'm going to be closer to the red light. Sorry. That's the value of this approach. That we can go to the cells, study that visual perception all the way, make predictions, and test them through a behavioral experiment. So now the model takes a very interesting role in terms of, well, how can we imagine these enclosures for the Peking ducks? And we're not there yet. So these are all preliminary findings, and so we need to sort this out, right? So what you have in front of you are the results of chromatic and achromatic contrast from the Peking duck's visual point of view. So what you have on top is the object, and what you have here at the back is the background, right? So this would be the blue feather against the ball, the wall, the duck feed against the blue feeder, and so forth, right? And you can see that the patterns are not necessarily the same in terms of the color signal versus the grayscale signal as they come to the brain, right? So if your question is, well, how do they sort that out, these two signals? We don't know the answer to that yet, right? Hopefully the behavioral experience will help us navigate that. But there are some interesting things here, right? So under the red light that we have right here, we can see that the blue feeder against the wall could be very contrasting. So by the way, this is the 4 GND threshold that I mentioned to you before. So whatever is below or close to that threshold is going to be very difficult for the animal to distinguish. Whatever is above that threshold is good news for the animal, right? But there are some interesting things, right? Like look here for the achromatic contrast, the duck feeder against the red feeder, right? Look how close under the red light that is, right? So in principle, based on that behavioral experiment, we should say, well, let's put red lights everywhere. That's a good idea, but maybe we need to sort of tune that. Maybe feeders need to be illuminated with another type of light. Maybe some portions of the enclosure require a different type of light. So that's something that we're going to investigate behaviorally with the predictions of these models. All right, so we're taking a very similar approach in the HENS project and many other things. As I said before, we don't have I don't have the time to share today, but we're seeing some interesting results. This is just the sensitivity of the visual pigments in the brown hens and white hens, and we are seeing differences in sensitivity, which might affect potentially their behavior. We're also doing a collaboration relative to dogs, and this is with a former grad student of mine, Brett Moore, who is an assistant professor at the University of Florida, right? It's also from the perspective of welfare, trying to understand their binocular vision, right? And we're relating that to their head morphology, right? So we're seeing that, for instance, brachycephalic dogs tend to have a wider binocular field than dorycephalic dogs, right? So we're trying to understand how that could have implications for the training of dogs and a few other things. So that's some of the aspects that we have been working together, trying to bring these two approaches into, or these different approaches into animal welfare. So now I would like to switch gears a little bit and try to talk about some other opportunities from the perspective of quantitative ecology that are more related to the way we do research, right? So I don't know if you heard about this, but in some areas, there is a lot of discussion about the reproducibility crisis in science. So is there a reproducibility crisis in science? And the answer seems to be yes, through many surveys and through a bunch of different resources that are out there, right? So apparently the way we're doing science leads us as a community not being super reproducible, right? And this stems from a study that was published in 2015, so many years ago now, in which psychologists decided to pick about 100 studies, seminal studies in the psychological sciences. Many of these studies were in textbooks, right? And decided to replicate. Okay, so they did that, and they found out that they could only replicate the results of 36% of the study. The other studies completely different, the other replications completely different results. And that led to a chain reaction in a bunch of different STEM disciplines that found similar results. The latest one is in cancer biology, and the results are very similar. Only 46% could be replicated, right? So maybe we should start defining what is research reproducibility, because that could be a concept that has different facets, right? So you can have methods reproducibility, which means that based on the description of the methods in your paper, another scientist comes, reads that, and is able to reproduce exactly the methods that you have used, right? You can also have results reproducibility, right? in which somebody, based on the description of the methods, run the study and gets a similar result. Then we can have inferential reproducibility. What that means is that the original data set is given to another research group, they run the analysis, and they get the same statistical result. And then we can have this generalizability, which basically is somebody else runs the study again from scratch and gets similar results. So there are different levels when it comes to reproducibility, but they're all part of this research reproducibility crisis that we're talking about today. So the important thing is that reproducibility is closely tied to two other concepts: the responsibility we have as scientists to be very detailed and to be able to make sure that another scientist could replicate our methods and our results, which is one of the tenets of doing science. And the idea is that if these two things are working together, we're going to increase the rigor of the research that we do. Okay? So in ecology and evolution, unfortunately, this lack of reproducibility crisis has taken a very dark side, because there are multiple cases of very famous ecology and evolutionary biologists that committed fraud, basically, right? And you know, this is extremely unfortunate, and it is something that in our field we're still coping with. Particularly, you can imagine the negative effects for students, right, and postdocs and early career researchers. But at the end of the day, whatever way a study is not reproducible, irreproducible biology costs about $28 billion a year. And that's a big deal, right? Because it's a waste of the taxpayers' money and undermines the public trust in science. And today, we need a lot of trust in science, as you all know from the news that we are getting, right? So this is not a good thing. All right. So what are the approximate causes behind the reproducibility crisis? When I ask that question, right, something weird happens, because the first answer that I usually get is fraud, of course, right? Oh, those people that commit fraud, they're awful. They are the ones causing the reproducibility crisis. Actually, they're not. So some studies have measured, which is very difficult, very challenging, actually, the incidence of fraud. Apparently, it's very, very small. But people think that fraud is the driver behind the reproducibility crisis, and then some people might think, well, I don't commit fraud, so I'm good. So actually, the main cause behind the reproducibility crisis is a series of common scientific practices, right? So let me mention some of them. Underpowered studies. So what is an underpowered study? It's a study that has a sample size lower than needed, think of a power analysis, to run and detect a certain effect size, right? A lack of understanding of what a p-value means and what effect sizes mean. A lack of within study replication, lack of understanding of core statistical principles. A lack of statistical reproducibility, meaning here's a data set, I just analyze it, you go and analyze it, you do, and we get completely different results. Errors in experimental design, in randomization and blinding, in sample size estimation through power analysis, in the interpretation of the statistical result. So we show two groups the same result, they interpreted it completely different. This also leads to biases, like Harkin and p-hacking. So Harkin is coming up with hypotheses after you see the results, which is a bias in the literature that we're trying to get rid of. And p-hacking, which is you run a bunch of analyses and you cherry pick the significant ones to present in the literature, right? So at the end of the day, this is driving the reproducibility crisis, not fraud. And that's very concerning because we have these common scientific practices all over the place, unfortunately. Okay, so we also need to understand that reproducibility problems can occur at different levels, right? they start with each of us. Example, today I run a statistical analysis, and then I have other projects, I forget about it for 2 years. 2 years from now, I go back to that file and go, what? And it's me who ran the analysis. I don't even know what I did. What's going on? I don't understand. So that's step number one. Step number two, within labs between individuals, right? A student graduates, we have a data set, right? A new student comes in, hey, here's the data set, go analyze it. And the student goes, I don't even know what's going on here, right? And of course, between labs, right? So as we go up the ladder, right, the probabilities of accumulating errors goes up and up and up and up, and that's a big problem. The other problem is that we used to think that peer review was the excellence point, right? So we got peer review, we got accepted. This study is perfect. It got accepted. It got published. But nowadays, the value of peer review has been diminished quite a bit for multiple reasons, right? It's difficult to find peer reviewers. But the most important reason is that we are using more and more complex statistical analysis. And we don't have a community trained on evaluating this complex statistical analysis. So the reviewers are making bad decisions in many cases. And many papers are allowed to be published when they shouldn't be published. So that's one of the crises we're dealing with. So now we have to deal with another problem, which is these different groups. One of them is top peer, right? Which what they do is they peer review after publication. And what they do is a paper is published. They say, okay, let's analyze the paper. Let's do. Usually you have to share the data these days. So they get the data, they reanalyze. And if they find some issues, they go and post it in this paper thing. And if the issue is serious, they contact the journals. There is a system for them to contact the journals to start the process of retraction of that paper. And this has nothing to do, I shouldn't have said that. Fraud, the incidence of fraud here is very tiny. It's mostly related to these common statistical practices that I was telling you about before, right? Skip this. So this increase in the paper sort of sites has increased the number of retractions in papers in an amazing way, and this keeps going up. Okay, and most of these retractions are not because of fraud, are because of these common statistical practices or mispractices, right? All right, so this crisis has led to the proliferation of new quantitative approaches, particularly coming from those fields that started the reproducibility crisis, like the psychological sciences and other fields, to try to better design and analyze data. And also, it has led to the revision of classic approaches to ensure the proper application of statistical principles, right? So there are many things coming up in the statistical literature, new things, right? That people like me, who have trained decades ago, are completely unaware of. So the way to think about this is think of the literature in animal sciences now versus 40 years ago. Has it changed? Tremendously. Well, the statistical literature is the same. But we're using, in many cases, statistical tests that we learned 40 years ago. You see the problem? So I looked a little bit, not too much, into the animal welfare literature, and the really good news is there are some papers that are starting to question this issue of reproducibility. So this is really good news, right? So this is very encouraging, right? So what I want to wrap up the talk today is talking about these three things, very tiny things in the big context of the reproducibility crisis that can help us deal with some aspects of the reproducibility crisis, right? So the first thing is share. So that means that when we submit our manuscript, when we submit, not after it gets accepted, we should be sharing the data. We should also be sharing the code that we use for the statistical analysis at the moment of submission. And we should also ensure that the statistical analysis that we run and the figures are computationally reproducible, meaning that we ask somebody else to run that code for us. And, hey, can you make sure that you got the same results that I got, that you can generate the same figure through the code that I generated? That's very important, right? And what that means is that R or Python are the platforms that we should be switching to in order to ensure that reproducibility, right? I've heard that some people use SAS a lot, which is good because I grew up in SAS, so I know that. But the problem with SAS and the reproducibility crisis is that not very many people have access, financial access to buy the SAS license. So if you're in an academic environment, that's okay. But some postdocs move to industry and they don't have access to SAS, right? So then they cannot reproduce the code. And that's where R comes in, right? The other thing is that the SAS functions are hidden because of a proprietary thing. In R, all the functions you can see and exactly know what the person who built that package did in order to get a particular output. So that's another advantage of R. So, but the share argument goes beyond being reproducible. That argument, some people are saying, well, maybe, and just maybe, if we share all these, maybe there are reviewers out there that are going to download the data, download the code, go through that, and say, oh, I know what you did, and I think you just need to modify this. And the argument is that maybe by sharing all these, we might increase the chances of getting a paper accepted rather than decrease the chances of getting a paper accepted. Because if you're a reviewer and you say, I use general linear models, period. Oh, okay. Versus your reviewer say, I use general linear models, the code is in the appendix, the data is da-da-da. What I do, I run that and go, yeah, well done, perfect. I know exactly what you did, right? So that's an argument to think about. The second thing is that some statistical analyses are very complex. And if we lack a deeper understanding of core statistical principles, then there could be problems with the misalignment between the data and the analysis setup, errors in running the analysis, a misinterpretation of the results, and even an overgeneralization of those results. So what I like this thing. So what seems to be happening today is that researchers, they're trying to learn everything related to machine learning and AI. Tell me, I need to learn. But they don't know the basic core principles, statistical principles, and how to apply them correctly. Right? So this is a big problem that we have these days. To exemplify that, I'm going to use the case of mixed models, right? So mixed models are very complex models. Here is a schematic representation of the components of a mixed model, right? And the idea is that if you're running a mixed model, you should have a pretty decent understanding of all these components in order to be able to set up the model correctly and interpret these results correctly, right? However, that has been a challenge. So people are using mixed models like crazy. Mixed model this, mixed model that, da, 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 da. But they are not recognizing those challenges. In fact, a few years ago, this paper went back and looked at the mixed models published in the literature and realized that many of those mixed models had issues in the definition of the within subject factors. They were ill-defined in the model, and consequently, the results of many mixed models in the literature are incorrect. Right? And also there are other papers that say, well, the way we define our random effect structure needs to have some principles rather than I'll just put things in. So there are consequences of the variation in the implementation of the mixed model. So a couple of weeks ago, we published a paper in which what we did is, okay, we had two data sets, and we said, here's the question. So we For each data set, we have a very specific question, and we ask for volunteers, different groups around planet Earth, to analyze the same data set with that question. And we said, you independently analyze that, but don't talk to each other. Just do that. And the results were shocking. The same data set, the same question, completely different statistical results. So one of the things that this paper identified is that we are not applying core statistical principles in the same way. And some people are not applying the right principles, right? So again, this is an argument for moving into R, right? Because we have a transparency in understanding how R is applying those principles, right? And that's very difficult to do in SAS. So at the core of this problem is a mismatch between the way we practice statistics, or we should be practicing statistics, and the way we're training early career researchers in statistics. So my point is that we really need more formal and informal training, right? And that could take different shapes and forms. New courses focus on difficult statistical methods from applied perspectives, lab meetings discussing code, local coding clubs, seminars on new development on existing statistical methods, groups providing guidance on how to improve the statistics from grant proposal applications, and so forth. So that's something for all of us as a community to think about. The final thing, which is interesting, is the correct application of a p-value, right? So we have all run statistical analysis, right? And we're like running the analysis and kind of hinting at that p-value that please be less than 0.05, because then my life will be perfect, and if it's higher, my life will be off, right? So that stems from the null hypothesis testing framework, right? Which basically says, well, there is a threshold relative to the null hypothesis, right? If your p-value is above that threshold, then you fail to reject the null hypothesis. Basically, you got a non-significant result. And if the p-value coming out of a statistical test, let's say it's this one, is lower than the threshold, then you reject the null hypothesis, meaning that result was statistically significant. Okay? So this is important because it leads us to the proper definition of a p-value, which is the probability of observing the collected data in a given sampling event, not in 35 sampling events, in that sampling event, assuming that the null hypothesis is true. So the p-value is not the probability the results have occurred by chance. That's not what the p-value is. This is what the p-value is. So my hope here is that the p-value is the p-value. It's a tiny little thing. So that's one concept. The second concept is that the p-value does not exist in isolation. The p-value exists related to these 4 elements, which are mathematically related. So the power of a statistical test, which is 1 minus the type 2 error, and the type 2 error is the probability of a false negative. The significance level, which is the type 1 error, which is the probability of a false positive. The effect size, the magnitude of the difference between a treatment and a control, whatever, and the sample size. So think of these elements, I'm sure you have seen them in power analysis that you may have run. So these 4 elements provide the space of inference. So you get a p-value of our statistical test, you look at it, but you say, okay, let me wait a second, and then you put that p-value in the context of this space of inference. That's how you interpret a p-value. Okay? So the implication is that, I got a significant p-value. Well, that significant p-value might be a fluke if you have a high type 2 error or low power in your statistical analysis. If your study sample size is lower than the estimated sample size coming from a power analysis, and if the observed effect size is larger than the expected effect size coming out from a power analysis. So my point here is that, I got a significant value, I should submit the paper to sign. No, wait. Before you do that, I mean, if you want, that's great. But before you do that, you need to look at the context, at that space of inference, right? So some solutions are to run proper power analysis. One of the things that we're realizing from the statistical literature is that when we have very complex statistical analysis, the usual power analyses are of no use. And we have to use other approaches to run those power analyses. And the approach that seems to be the best these days is using simulation, right? We need to interpret the p-values in reference relative to all the other components that I mentioned to you before. And we should report not just the p-values, but also the effect size, right? So how much of a difference we find between that treatment or control, or how strong a relationship seems to be. And with that, hopefully, I've given some free of opportunities for us to think about in terms of collaboration. Thank you so much. Question? Yeah.
Well, great talk.
Thank you.
And so one of the problems with your reproducibility that I see, so I'm a section editor for poultry science. I think in the last 2 years, my number is 550 papers that I've evaluated or been in charge of evaluation. And I tell you, a trend that I see coming from around the world, very much related to what you're talking about, is where people are doing an experiment, and then literally, and I've counted them, measuring 60, 70, 80 different variables from completely different aspects of life.
Yep.
And seeing a half a dozen that are significant and literally connecting the dots.
Yep.
And saying, this is a paper.
That's a huge problem.
Yes, it is a huge problem for me.
Yep.
So, you know,